So thank you very much for having me here today after a year delay. It's virtual, but still, we're here. My name is Fred. I work for Inari Agriculture, which I will introduce in a minute. I am the Vice President of R&D there. And I'm also happy to be here because the Bayer cross is actually very familiar to me. I worked for Bayer for a number of years before I joined Inari. And today I'm going to tell you a little bit about how we are using machine learning models to solve a number of problems that we have so that we can get to our goal, which is basically addressing some of the global challenges in agriculture. What I'm not going to do today is I'm not going to go into detail and depth of how it works, but more the application for two reasons, because I think that's more in the spirit of what this conference is about. Also, as a disclaimer, I am not a machine learning specialist, but I'm more interested in how we apply those kind of tools on very specific problems in agriculture. So with that, we can go to the next slide. So Kathy is controlling my presentation for those people who are asking. So this is a little bit the outline. Who we are, what Inari needs to do, and then what kind of technology we want to use for that, and then how we do it, which is basically applying some of the predictive modeling to some of our problems. All right, next slide. So very briefly, Inari has been rebranded recently with the new colors that you can see here. We call ourselves a seed design company, which basically means that we are reshaping genetic diversity in different crops to help farmers and also help to build a new food system, basically by including technology for gene editing plus predictive design. So, or to put it the other way around, we use predictive design to know what kind of modifications we need to make for certain traits, and then we apply gene editing to change those, and also some other genetic advancements. And then we hope to have really important step change products, as we call them, with the major impact on what the plant in the field looks like and can do, so that we can also build with all the other players to a more sustainable future for the food system. And you'll see why, because we work on very specific traits that are linked to this sustainable and sustainability goal. So in the next slide, just at a glance, who we are as Inari, for people who haven't heard of us. We were founded in 2016, which is not that long ago, as a flagship pioneering company. Some people might know Flagship Pioneering. They're one of the big investment companies that, for example, also invested in Moderna, who everyone knows now, one of our sister companies. We're headquarters in Cambridge in Massachusetts, so near Boston. And then in 2018, we went through an expansion where we started a site in West Lafayette, which is in Indiana. It's a production site where we do a lot of upscaling of our seeds. And then we also started a site in Ghent, which I'm heading. And this is mostly an R&D site where we do a lot of research on plants, traits, and also some of the computational work that I'll talk about. And so in our leadership team, we have people from all sorts of different companies covering the industry, but also some other companies. And then you can also see quite some diversity also in our staff. So we have all kinds of scientists, not only plant biologists, but also physicists, chemists, data scientists, engineers, and so on. We're about 180 FTE right now, 180 people, representing both people coming from academia, people coming from agriculture, and then from the non-ag. Okay, next slide. We also have a very strong scientific strategy board, which is essentially an advisory board that helps us with many of the scientific challenges that we have. George Church, which I hope a lot of people know, is a professor at the Harvard Medical School. He's a scientific co-founder. Steve Jacobson is professor at UCLA, also scientific co-founder. Rodolphe Baranjou was involved in some of the early CRISPR-Cas work. And Jennifer Doudna, I think most people know, won the Nobel Prize this year. Dick Flavell is based in the UK and was the head of the John Ennis Center. Dirk Kinsey is the head of plant PSV at VIV, and Zach Lippman is at Cold Spring Harbor. And Jonathan Pritchard is a population geneticist, which helps us a lot with computational work. All right, so next slide. So what do we want to do? Like I said, we want to transform the food system, which is basically, you can click on Catherine for the animation. Thank you. So essentially, in the blue box, you see what we want to do, right? So the seeds are key for us to solve some of the issues with the food production throughout the world. I mean, growing population, more demand for food, more demand for feed as well. Also, farmers are sort of locked into what they can buy from a number of companies. The market is not very diverse. And then how do we see our role? Like I said, we want to leverage some of the new technologies, CRISPR-Cas, that I'm not going to go into today, to unlock some of the genetic potential of seeds in the different crops. And the kind of products we work on, as I said before, are linked to sustainability goals. So we want to develop plants that use less water for the same yield, that use less fertilizer for the same yield, so that farmers can use this, have still the same yields or even better, but that there is less strain on environmental and environment. And then we also work with partners to have a seed network to sort of diversify a little bit the seed market right now, so that it gets accessible to most farmers. Okay, so next slide. Yeah, this is just an example of what happens for example, for soy. We also work on other crops, but this is just an example for soy. The main problem that we have is that yield has been increasing a little bit for soy, but not as much as we would like it to have, right? So we want to go to up to 20% of yield increase, and then also therefore create more value for the farmer, and because of that also reduce the amount of land that's needed for soy production, which is very land intensive. Okay, so next slide. Okay, so how do we want to do that? So sorry, go back to the slide. Yeah, so this is kind of how the evolution was. When I say breeding 4.0, it's basically what happens is over the last hundreds of years that we've done farming, we've had stepwise increases in output. Some of that is because of practices in the field. Some of that is because there is new technologies that's being used, and then there is a whole bunch of emerging technologies that we're exploiting, which will help us to make the next step to the next level. Some of that is including breeding timelines, accelerating breeding timelines, increasing the diversity of the genetics of the crops so that we can do more different kinds of traits, and then improve basically crop performance. Okay, so next slide. All right, so now we're getting to the bit that I want to talk about most. So essentially, our approach is built on two main things, as I said before. So in the blue box, you see how that was done until we had those emerging technologies. So legacy approaches, not that there's anything wrong with them, but just like this is part of the technological evolution. Where in traditional breeding, for people who don't know how that works, is essentially there is a lot of trial and error, right? Breeders make different kind of crosses between different plants. They put them in the field, they select the one that they like, and then they continue, do the same thing the next year. So this has worked very well for us as humanity, because this is what we've been doing since the dawn of agriculture. But the increase of yield is quite limited this way and quite slow. Right? And then we had GMOs, which are an improvement also. They do help you to increase a little bit and add new traits that you couldn't do before. But they've been mostly used for pest control solutions, because you can only put in one gene at a time and so forth. It's also very lengthy because it's very regulated and expensive. So what we want to do is a shift from that. So on the lower bottom, you see the multiplex editing, which I'm not going to talk about today, but this is essentially, we have tools that allow us to modify plant genomes very quickly and efficiently. And basically we do what breeders do, but we do it much faster, right? That's one part of the equation. The other part of the equation is if you have the tool, the scissors to do it, you also need to know to have the knowledge of knowing what you need to change. And that's what I'm going to talk about in the next couple of minutes. This is basically the predictive design. So there's lots of genes in the genomes. They all do something, or most of them do. But the trick is to figure out which ones do you need to modify and how in order to develop the traits that I mentioned before. For example, reducing water use or reducing need for fertilizer. Okay, and so this is part of our breeding design. So in the next slide, you will see four main problems that we're trying to tackle in order to achieve the higher objective, which is improve plant performance. Right? I'm going to briefly touch on all of those. If you have any questions later on, I'll also be in the breakout, so we can talk about further if you want. So the first one is the computational breeding at the plant level. So remember I talked about how how breeders just take two plants that they like, they make a cross, and then they look at the offspring and they take the best one and they continue on. So this has been the way we've done it for a long time. We also heard yesterday some one of my colleagues from KWS who mentioned genomic prediction and new kind of approaches to do computational prediction to select which plants. Which plants you need to cross before you actually cross them, right, based on genetic data. So that's one thing that we also do. I'll talk about it. Then there's the other element of gene and network level. So this is really about discovering and mapping gene networks for complex traits so that we can basically understand better how genes interact and then how we need to change them. Then there is a third component, which is really looking at the overall genetics of the plant. We want to identify all of the major small changes, which individually are not that important, but taken together have a major impact on the plant performance. And we are developing tools also based on computational filters to predict which of those we need to change. And then last, but definitely not least, we also use deep learning models to design optimal protein structures. So these are protein structures that don't really exist yet in nature because evolution is taking a certain path. And we are redoing evolution over and over again in silico so that we can have novel proteins based on the original protein, but that have improved functionality. Okay, so I'll briefly touch on all of those in the next 10 minutes. All right, so next slide. Okay, so I just have this one slide as because I don't have time to go into how it works, and nor would I be the right person to do so. But it is kind of important to understand how we can apply this to some of the problems that we face. So machine learning is a very large term that can cover also different methods, right? Essentially, it is all about building models, right? And so there's different ways of building a model either through supervised learning or unsupervised learning. So key to that is that you start from biological data layers, which can be multi-layered. So as you can see here on the left-hand side, so these can be genetic data, sequence data, epigenome data, transcriptome, metabolome, proteome, so multi-dimensional data sets, which are getting pretty abundant now and not too expensive to generate, and adds a lot to what we call the molecular phenotypes, right? So these are the biological data layers. And then there's also information that we can integrate on phenotypes. So this is basically how the plant looks like, either in a greenhouse or in the field. We can make measurements, take images, all sorts of readouts on the plant level, which we can connect also to either the genetics or the transcriptome of the plant. So these are the so-called GWAS and TWAS data sets. And then we can also integrate what we call biological priors, which is essentially information about genes that we already know about, physiological processes that have been characterized. So this is active information that we can integrate in such a model. And then on the left-hand side, the right-hand side, you see a very simplified overview of how such a model can then be built and tested also, and then the differences between either you do this through supervised learning, where basically you already have you try to obtain a model which maps predictors to specific target variables. For example, you have DNA sequences, and you use that to find histone marks, or the other way around. And then you have unsupervised models, and this is where things like artificial neural networks and variants of that come into play, where you don't have such a specification about the outcome in the data sets. So that requires a lot of training, a lot of validation. There's some issues there that I don't have time to go into, which are not necessarily linked to the fact that this is for biological output, but just with machine learning itself, that you can overfit or underfit and so forth. Bottom line is that it's really important to be able to do the right testing, but also the data. That was also something that was mentioned yesterday, I believe, is that the data challenges are also there. Data needs to be high quality. So I put quality, quantity, so sufficient quality, sufficient quantity, the right type also, and the diversity, which is basically the right type. In plants, this is more a challenge than, I believe, than in humans, because of course plants are very diverse. Humans are not. It's one species versus many species. So that in our experience also, that makes it sometimes quite challenging to obtain data sets at the same diversity and level than we would have, for example, from an ENCODE project in humans. There's another challenge, which is also there are a lot of tools out there to apply on data sets for machine learning approaches. Typically, they're developed for human or mammalian research, not necessarily for plants, which means that we need to convert them to that, and so that requires a lot of work. I put a reference here for people who are interested. This is a paper from the virtual lab. It is a really good paper. I recommend it to people who would be interested. Okay, so that's for the primer here. Then next slide, I talked to you about, yep. So the first problem, we heard a little bit about that yesterday. This is one of our issues is computational breeding. So like I said, breeders until recently went pretty much based on their preference, visual preference of a plant with another plant that they liked, they crossed them together and so forth. Now we are already a couple of years in the next sort of level where we use genomic selection and also envirotyping. So this is also important. Genomic selection is essentially you look at all the sequences that you have from all your potential parents in your population, and based on your model, you will select the right parent combinations because you can predict the performance of the offspring, right, with pretty good accuracy. This is really valuable. Why? Because it avoids having to do many different crosses, actual physical crosses, which are very expensive to do and take a lot of time. So this is why this is so important. The envirotyping is another additional layer, which also looks at the fit not only of the genetic background of this specific parents, but also with the environments, which can be very different. Some genetic backgrounds are very compatible with one environment and others are not. So this is also information that's integrated into this model. And then we have things like pedigree viewers that we can use to also look at the relatedness of different lines. And then also tools, haplotype tree, which basically allows us to look at our entire germplasm and see how which blocks of chromosomes and DNA are shared across our the members of the germplasm. The output is that we have, in some cases, we have between 70 and 94 prediction accuracy for yield, right? So this is really important. And this is all because of the modeling part, right? This is really a breakthrough because that means that not doing any cross, most of the time, you will be able to predict which parents you need to combine, and it will turn out to be true if you actually make them in the field and you test for performance. This is a major game changer compared to how we did it, 20 years ago. And that will help us to increase also basically the best combinations and increase the output and performance of the crops in the field. But it's not the only thing. So this is one component. The next slide, you will see the other problem that we're trying to tackle, which is from a very different angle, where essentially instead of looking at the whole genetics, we use modeling to understand how genes interact together in a network. So in a nutshell, plants are composed of obviously different genes, but also different physiology. And what we do when we look at a specific problem, like nitrogen use efficiency, so this is the use of fertilizer, we try to break it down to very specific physiological components. And then we use cell models as a way to do in plant testing, but also coupled to computational models to be able to predict how those genes that we know interact and then identify novel genes, which we might then modify in the same kind of approach to refine our model, to then have as output very specific genes that we say, okay, these are the ones we need to modify to have a plant that would be more efficient in the use of nitrogen. So we have done that on the right-hand panel. That's less about the computational part. That's more about the fact that we are actually doing that in very high numbers, so many, many genes at the same time that we're modifying. And this is in collaboration with our partners from BIB, where we do a lot of phenotyping and genotyping, which we then also will feed back into the modeling that we do at the initial phase. So this is a very important part. So that is still very much ongoing, but very exciting work that's being done here again for the most part. Then in the next slide, you will see, well, this is more of what I said. So this is also a little bit more into detail. The molecular phenotypes I talked about earlier are essentially, we use them for prediction of expression values of the genes. So before we actually measure them, so we can say, okay, this gene is expressed at this level and this gene will be expressed at that level. And we would like to use that also to help us to identify what parts of the regulatory part of a gene needs to be modified to have a specific phenotype of the plant. For example, more seeds, bigger seeds, and so forth. Okay, so next slide. This is the third component where we try to unlock all of the genetics. So very dense story, but I'll try to do it in 1 minute. Essentially, we already said that crop yield are plateauing, right? Especially in corn, meaning that they're not accelerating as much as they used to for different reasons. We are looking at identifying point mutations, so point genetic changes in the maize genome across many, many different individuals. That individually, those changes have no impact on yield, but if you add all of them together, they do have a major impact. The challenge here from a computational point of view is, what are those targets? What are those single nucleotide polymorphisms that we need to modify? This has been the work of a couple of years now, where we developed very specific computational filters, which I don't have time, unfortunately, to go into, but that help us to provide an output that is then used with our multiplex genotyping machinery to then make the changes, which is what we're doing now in the plant, and then have the hopeful improved output, all thanks to the model. Right? So then the last element in the next slide. Yeah, I'm going to skip this one because I don't have time, but yeah. So this is what I mentioned before. This is kind of an extended view of how that process works. So we assemble data from many, many different genetic variation. We prioritize the sites using computational filters, and then we use neural network-based machine learning. As I mentioned before, biological priors are also used, and then the tools for personal genomics that we've adapted to plants. And then we want to edit all of those genes to validate our hypothesis, do the editing also in planta, and then have plants with improved performance. So this is where we're at right now. And then lastly, but not least, so this is a really important application as well that I mentioned in the beginning. So we are also applying deep learning methods to re-engineer proteins. So essentially, we have, everyone has heard of Alphabet's DeepMinds, which is one kind of application. They use that for predicting protein folding. We are using what we have developed for improvements of the protein function, which is a very different thing. And so we want to use that to redesign our own proteins that we use for modifications. Very simple, I mean, relatively speaking. But we also want to use that for new proteins that we've identified or proteins that have been around for a while, and that we can improve using those deep learning approaches. And then in the next slide, it's just my penultimate slide. You will see, if you can go to the next slide, Kathleen. Yeah, so this is, I just showed it because it's so cool that I had to show it, how it works. So essentially what you see here on your left-hand side, so those hills, this picture landscape represents evolutionary space, right? So these are all the possibilities of which form your protein can have. The green dots on the mountain there are what the track that evolution has chosen. So these are the existing versions of a protein in all organisms that are alive on this planet. In order for a protein to evolve to something very different, it would have to go to a stage where it's actually less effective than it is right now. And then through evolutionary processes, it would not be selected. So you can never get to the other hill that you see there on the right-hand side. So what we want to have is we want to have a protein which is on that higher peak there, so the orange bar, the orange dots. But evolutionary, you will never get there. So the only way to actually make it is by exploring all of that evolutionary space using deep learning approaches, and then come out with the one that is predicted to be at the higher performance level, so the higher peak than all of the others. And what's really so cool about this is that this is something that would never have been generated through evolution right now, but it is the same protein and an improved performance. So there's some papers that have been published by, among other people, are scientific advisor George Church, who have done that on a green fluorescent protein. And we're now applying that also and improving it and applying it on many other proteins. So very important development there. And then I'm going to skip this one. It's just some examples. So this is essentially what I talked about before, how we can use this application then to improve the speed and the cost of improving those proteins, which is going to help us a lot with our projects. And then just for the sake of time, I will end my next one. Slightly over, apologize about that. So I thank you for your attention. I know this was pretty dense, but there's a lot of cool things to talk about when it comes to applying machine learning and modeling into our field of work, because this is a very exciting time to be working in this kind of technologies. So I thank you. I will be also for the questions later on available if you have any.